Hi all, so good to have everyone here. Good to see you guys in person and people on Zoom, obviously. My name is Malvika, and I'm a third-year PhD student in Dr. Fowler and Dr. Starita's lab in this department that's hosting the symposium, Genome Sciences. And so I wanted to talk to you a little bit about the data we produce in lab and how we can use that in the clinic for patients. To start off, I wanted to reorient you guys first to the central dogma. So DNA is transcribed into RNA, which is translated into protein. And proteins are kind of the workhorses of our cells, right? So they have a specific function that they perform, and they help the cell live and grow. Oftentimes, though, as we've heard from the previous two talks, we have variants in our DNA. And so these are like sometimes single nucleotide changes that can then produce, for a specific protein of interest, a slightly different protein. And these slightly different proteins can sometimes be disease-causing, which means that this protein is not functioning the way it's supposed to in the cell, and therefore the cell can either die or just grow really slowly. Or other times, these variants for these slightly different proteins can be not disease-causing, which means that they're functioning the way they're supposed to be. Other times, though, we have these things called variants of uncertain significance, as Stephen had mentioned earlier. And so we're really unsure how they perform. So how is this protein doing in comparison to what it's supposed to be doing? And so what does that look like in reality? Let's take, for example, the BRCA1 gene, which is a gene that is important in DNA repair. And so if we have a variant that's performing well or as it's supposed to be, then this variant can fix the BRCA1, can fix the DNA repair mutation in your DNA, let's just say. Other times, we can have a disease-causing variant, which means that it cannot fix this break in your DNA, which can lead to breast and ovarian cancer, specifically for BRCA1. And last but not least, supposing you have a variant of uncertain significance in the BRCA1 gene, we don't really know how it performs. So it could be okay. So it could be, green, can fix the break in your DNA, or it can lead to breast and ovarian cancer, just increasing your likelihood of that. And unfortunately, this is kind of a big problem in clinical genetics. Basically, 90% of missense variants, meaning 90% of variants that have this single amino acid change in a gene, are variants of uncertain significance in this big database that we have where we're collecting data about variants and if they're disease-causing or not disease-causing. And so how do we actually try and resolve these variants of uncertain significance? Well, we need different kinds of data for that. One of the most important types of data can be patient phenotype, which means that let's say a person comes in with a variant in the BRCA1 gene, and we see tumors in the breast and ovarian tissue. There is some type of evidence telling us that this person's variant in the BRCA1 gene is disease-causing. Next thing we can do is we can use family history. So we can actually just check their family. Is this variant segregating with the people who are affected with, breast cancer or ovarian cancer? Next thing we can do is use population frequency. So we can look at the population and we can say, how often do we see this variant in the BRCA1 gene in the population? If we see it at a very high frequency, we kind of know that it's not disease-causing. It's most likely not disease-causing, right? Next thing we do is we have predictive models. So researchers have worked really hard on this, where they can actually use data and train models to predict these single amino acid changes and how or whether they're disease-causing or not disease-causing. And last but not least is we can actually just generate data in lab. So I'm calling it functional data because we're kind of understanding or assaying how each variant performs in lab. But you can also say it's lab data in this case. I'm using it interchangeably. And so what is functional data or lab data that we generate? What does it look like, right? And how does it help? So let's take, for example, an individual that comes in, right? And we know that they have a variant in the BRCA1 gene. Well, we can just take this variant actually and introduce it into cells. And BRCA1 is actually essential for growth. And so if we have introduced BRCA1 into a cell, we can kind of just survey how it grows. So we can grow for 10 days and maybe the cell just dies and we don't see it anymore. So it's a disease-causing variant. That's kind of what it's telling you. Or we can survey for 10 days and maybe it grows really well. And so we can say that the BRCA1 protein is kind of functioning the way it's supposed to be, right? But as Stephen and Lizzie mentioned, there's 21,000 genes, right? And each gene has so many variants. And so if we were to do this for every single individual that walked into clinic, it would take forever. So we need to make this more scalable. And luckily for us, researchers have made it more scalable, right? So what we can do is we can take different variants in the BRCA1 gene, right? And at the same time, we can introduce these variants into cells. So just in a petri dish, in a cell, like in every cell, you can introduce one variant. And you can also have controls along with it. So you have previously classified controls, right? So you have these yellow cells out here that we know are okay, BRCA1 is functioning. And then we have these purple cells out here where we know that BRCA1 is not functioning, right? And then these other colors are just kind of telling you these variants that we don't really know what they're doing. And so once we kind of grow these for 10 days, we see and we survey how well these cells are growing. And so what you want to do is you just want to count. Basically, it's just counting, right? So we know our controls and how they're supposed to grow. So we see that the yellow cells out here have increased twofold, and then these purple cells out here have decreased twofold. And so what we want to do is basically extrapolate that information to the other variants that we've introduced into the cell line. And so the green and the gray, so the green has increased twofold, and so has the gray. So they're variants that are, not disease causing. And then we have this turquoise variant that unfortunately has decreased twofold. So it is a disease causing variant. And so what does that actually look like? How do we use this data now in the clinic? So we can have a patient that walks into a clinic with certain symptoms. And then the clinician says, you know what, let's get you sequenced. And so we get back their sequencing. We get back their DNA. Now, from this DNA, we actually have this variant of uncertain significance. We haven't seen this variant in the population before. We don't know what to do with it. So it's a variant of uncertain significance in the BRCA1 gene. However, I just showed you a functional assay that we performed in lab, right? We saw this gray variant before. We've seen it. And so when we surveyed this gray variant in our functional assay, we kind of understood that it's actually performing well. It's okay, it's not disease causing because the cells are growing fine. And then we can also look at something like the family history, right? We can look at the family history and see whether this variant is segregating with people who have breast and ovarian cancer. And maybe it's not, actually. So if it's this person, then it's not. And so what we can do with this data is we can just combine it, this functional data that we're producing in lab. We can do this for many genes, and many variants at once. And so what we can do with that data is kind of just go take it and tell people and patients, hey, you have a variant that is not disease causing. Kind of just informs their clinical decisions. It tells the clinician and the patient themselves that, hey, you don't need more, clinical surveillance. Or if it's a pathogenic variant or a disease causing variant, you can tell them that you need more clinical surveillance. And therefore you need to do, a couple more tests or mastectomy or something, right? And if it's a variant of uncertain significance, though, you can't really say that. And unfortunately, with no functional data or us not producing data in lab, that's kind of the reality of it, right? I told you earlier that 90% of the missense variants in that database are variants of uncertain significance. We don't know what they're doing. And so these are all actually just patients themselves that have no idea what they need to do next, right? But if we were to introduce functional data, we can go ahead and reclassify or tell patients the next steps that they need to do, right? And so what I want to leave you with is this feeling of like the data that we produce in lab, the technologies that we create in lab are really important, can be translated into clinic. And this can change lives, actually. And so with that, I just wanted to end the talk and ask acknowledge my lab, Fowler Lab, Sarita Lab, and the BBI, and give you some time for questions if anyone has them.
I think we could take like one question.
I don't know if this is just in this example, but do you typically only grow the samples for up to 10 days? Or is there like the duplication of it like being similar to the yellow ones?
Yeah.
Like what if it grows like beyond 10 days and now it's actually becoming more cancerous because it's growing too prolifically? So I'm just wondering if like time frame is accounted for, and how does that affect someone who is like in the clinic setting? Because time seems like it might be more sensitive, I should say.
Totally. Yeah, that's a great question. This was just an example Usually you grow cells at a certain level and you do screens basically that are you kind of like take your control variants that you know how they're growing, and you can grow them at different time points and just survey like how they're growing. And so, for example, if you take the yellow variants that are growing really well and you grow them for like 10 days, then 14 days, and then 26 days, and at 26 days, you survey that, oh, wow, these are just growing out of control. When you actually go to do your functional assay, you'd grow them only up to 10 and 14 days. So we do a lot of tests before we just, put variants in and start growing them. But you have a great point, because there are variants that will overgrow. Like we call them usually like in this setting, maybe like gain of function, variants. And so those are also cancerous, right? So it does give you a lot more information. If it's growing faster than wild type meaning like the normal like reference allele, it can give you that information. And so that's a great question. But we don't only use growth assays in the lab. We also use different things, like we can take a protein and we can fuse like a fluorescent protein to it, and we can see the stability basically of the protein. We can like sort, using this big machine that just sorts cells in a single basis. We can try and understand like how stable are these protein fusions with the variants. And so there's different ways you can assay how a variant performs in vitro. Yeah, a great question.